Hello students, if you are confused about the allosteric inhibition or allosteric modulations, this video will explain the entire modulation or allosteric inhibition process step by step in easy way. And this will explain in a simple, conceptual and exam oriented manner. So first of all, see what is the meaning of allosteric inhibitions. Actually, allosteric regulations, it is the enzyme has only one catalytic site where the substrate bind and another separate allosteric sites where the modifier binds. So allo means, what is the meaning of allo? Allo means other. So other site than the catalytic sites. Number two, allosteric and substrate binding sites may or may not be physically adjacent, and the binding of the regulatory molecule can either enhance the activity of the enzyme, that is allosteric activation, or inhibit the activity of the enzyme, that is allosteric inhibition. So in the former case, the regulatory molecules are known as the positive modifier or modulator, and in case of latter is the negative modulator or modifier. The binding of substrate to one of the subunits of the enzyme may enhance the substrate binding by the other subunits. This effect is said to be the positive cooperativity. If the binding substrate to one of the subunits decreases the avidity of the substrate binding by other sites, the effect is called the negative cooperativity. So in most cases, a combination of observed resulting in a sigmoid shaped curve. If you have to see the curve, or what we will find, this is the allosteric inhibition curves, and here the sigmoid curves in between the positive and negative modifier. So this is the Vmax, half Vmax, and substrate. So whenever we also increases the substrate concentrations, the graph shows first with the positive modifier or modulator positive sides, and the graph shows negative sides, there is the negative modifier when we add there. And otherwise, in normal condition, it is the sigmoid types or sigmoid curve. So there are various important characteristic features which we will discuss now. One is the inhibitor is not the substrate analog. It is partially reversible, but when excess substrate is added, Km is usually increased, Vmax is reduced. So the effect of allosteric modifier is maximum at near substrate concentration equivalent to the Km. When the inhibitor binds to the allosteric site, the configuration of the catalytic site is modified such as the substrate cannot be bind properly. So most allosteric enzymes possess quaternary structures, and they are made up of the subunits, like the aspartate transcarbamylase has the 6 subunits and pyruvate kinase has 4 subunits. So this is all about the allosteric inhibition. So students, if you have found this video helpful, please like, share and subscribe to our channel Physio-Pulse, and do not forget the press the notification bell so you never miss our upcoming lectures in physiology, biochemistry, biology. If you have any query, any question, any session, please give some comments in our comment section. Thank you. We will see you in the next lecture. Until then, keep learning.